So thank you for the kind introduction, and thank you actually for this opportunity to present some of our work. So the way I've sort of structured my talk today is in the first half, I'll sort of introduce the subject of CRISPR-Cas, but then sort of consistent with the theme of this session, I'll focus on some ongoing unpublished work in our lab, which we're very excited about, and which is in the realm of high-throughput biology. So beginning first with CRISPR-Cas systems. So these really are remarkable adaptive immune defenses that have evolved in bacteria and archaea that use short RNA to direct degradation of foreign nucleic acids. And sort of they come in multiple varieties. At least sort of 5 different types are known, of which type 2 is the best understood. And so here's an example of a type 2 CRISPR locus inside a bacteria. So it is typically characterized by a set of repeat regions that are adjacent to a cluster of proteins referred to as the Cas proteins. And just like the way we imagine an adaptive immune system to function, similarly, the CRISPR-Cas systems are also characterized by both an immunization phase and an immunity phase. So in the immunization phase, it basically arms itself, and in the immunity phase, it utilizes this information from the immunization phase to basically ward off a repeat infection. So the way that is believed to occur is, in the immunization phase, if this bacteria, and imagine this blue line is the outline of the bacterial cytoplasm, if this bacteria gets attacked by a particular virus, for instance, a phage, and if that phage injects a viral DNA into the bacterial cytoplasm, then a cluster of Cas proteins can process this incoming viral DNA and incorporate a small fragment of this viral DNA into the repeat spacer region. And in this fashion, what has occurred is that the CRISPR-Cas systems has now recorded a memory of this infection. So once this memory is now recorded in the repeat spacer region, one enters the immunity phase where one can take advantage of this information to ward off a repeat infection. And the way this occurs is that this repeat spacer region first gets transcribed into a pre-CRISPR RNA. This pre-CRISPR RNA, in turn, gets processed by a large protein Cas9 and a small RNA referred to as the tracer RNA, which sort of complex themselves onto this pre-CRISPR RNA to give rise to this final functional surveillance unit of the CRISPR-Cas systems. So this surveillance unit really sort of fundamentally comprises of the large protein Cas9 and the CRISPR RNA and the tracer RNA in hybrid with each other. So once the surveillance unit is formed, now if there's a repeat infection by a particular phage and that injects its viral DNA, then the surveillance unit can survey this incoming viral DNA. And if this information stored on the RNA matches the corresponding information on the viral DNA, then the Cas9 gets triggered to make a double-stranded break and thereby sort of destroy this incoming viral DNA. So it's really a beautiful system that nature has already evolved that sort of can record a memory of an infection and then utilize that memory to ward off the repeat infection. But really, the fundamental aspect and the most exciting aspect about the system is that the way it records that information and utilizes that information is in the nucleic acid domain. And sort of recognizing this aspect, we and others a few years ago engineered this system to also function in the eukaryotic setting. So just to sort of recapitulate, the CRISPR-Cas system really comprises of two components. It comprises of the large protein Cas9 and the small RNA that come together to form the Cas9 guide RNA complex. Just sort of looking at the nitty-gritties of the system, basically this is what it looks like. So you have the large protein Cas9 in complex with the guide RNA. So there's a constant region of the guide RNA, which I tend to refer to as the scaffold region, which is highlighted here in blue, that brings the Cas9 and the guide RNA together. And the region in green, which is referred to as the spacer region, basically provides the address to this Cas9 guide RNA complex. And then based on a homology search, once the docking of the Cas9 guide RNA happens, then the Cas9 gets triggered to make a double-stranded break. And similarly, just as it can be used to make a double-stranded break, the system has also sort of been expanded to also enable targeted genome regulation. And the way that occurs is one takes the Cas9 guide RNA complex and instead uses a dead version of the Cas9, which no longer has the ability to create a double-stranded break. But it still retains RNA-guided genome targeting functionality. And once you have this complex, then now you can recruit transcriptional activators or repressors or a range of other domains by either sort of directly tethering them onto the Cas9, or alternatively by attaching aptamer domains onto the guide RNA, which you can in turn use to recruit aptamer-binding proteins. So each of these approaches has now been sort of demonstrated to enable a host of sort of genome regulation and other novel functionalities. And the exciting aspect about the CRISPR workflow is that it is remarkably simple. All really one needs to do is to identify candidate target sites in your genome of interest, for instance, the human genome, and basically construct your corresponding guide RNA expression vector. And importantly, this can now be done sort of not just individually, but at the scale of large libraries, and then finally deliver the Cas9 and the guide RNA into your target cell type of interest. And I just sort of wanted to highlight one important point is that this is really truly a two-component system. And as long as one can express the Cas9 protein and the guide RNA in a cell type of interest, then one can trigger a double-strand break or a genome editing event. And that's the reason why this system has been very easy to port, not just across a range of cell types, but also across a range of species and organisms. And in many ways, I tend to think of Cas9 as the unifactor, sort of to take a Lord of the Rings analogy. It is really the only known system in biology, probably with the exception of the telomerase, that brings together all the sort of three classes of sequence-defined polymers, RNA, DNA, and protein in a programmable fashion. And that sort of really confers the Cas9 system an unusual amount of versatility. For instance, it can be used as a nuclease to make cuts and deletions in the genome and trigger homologous recombination. You can convert it into a nickase to make nicks or make safe double-stranded breaks by using an offset nicking mechanism. As I mentioned, you can take a nuclease null version of the Cas9, attach an effector domain onto it, and use it for targeted genome regulation or a host of other novel effector functionalities. And the same aspect can also be done by recruiting effectors via RNA aptamers attached to the guide RNA, again to use genome regulation or a host of other novel functionalities. And it's sort of important to realize that the system was really mined from nature. So the CRISPR-Cas toolset is sort of only growing very, very rapidly. And perhaps if not thousands, there are probably hundreds of thousands of Cas9 or Cas9-like single effector proteins out there in nature. And in fact, just looking at the existing metagenomic literature, there are already large regions in gray which we have in mind, and that just sort of, and that's only from what we have already sequenced. And if we start to sequence more and more bugs, we'll probably discover more and more novel CRISPR-Cas-like systems with novel abilities to target DNA and RNA in sort of unique fashions. And perhaps the most exciting aspect about the CRISPR-Cas systems is the fact that it starts to sort of enable the concept of human therapeutics, because the genome is now a druggable target, with the CRISPR being this very specific drug against any genomic fragment of interest. And at least in theory, one can start to contemplate the fact that one can start to eliminate disease alleles from the human population, or alternatively, even potentially engineer disease resistance. But if you sort of think about this paradigm a little bit more, then sort of a question mark very quickly emerges. This is because perhaps one of the greatest limitations to CRISPR-Cas for in vivo work is actually going to be delivery. But even if you sort of set aside the problem of delivery, which is a problem outside of the CRISPR-Cas field, and we just sort of focus on the broad classes of diseases, and we sort of classify them into the two broad groups, then you have sort of the classical Mendelian disorders on one end, such as cystic fibrosis and Huntington disease. It's important to note that these are very rare, but of course, because there are a few mutations associated with each of these, they are, at least in theory, targetable. But on the other hand, you have the polygenic disorders such as cancer, heart disease, Alzheimer's, which are typically characterized by a huge number of mutations, and the very thought of basically genome engineering it is really daunting. So how can one sort of go about addressing this paradigm? Well, one approach is perhaps we can take inspiration from nature, because nature can often solve a very complex problem by coming up with a unique solution. For instance, we have the CCR5 mutation in certain individuals that confers them the ability to have HIV resistance. Individuals with PCSK9 mutations seem to be resistant to heart disease to a certain extent. But perhaps the most interesting category in this list are supercentenarians, which in some sense are basically already have some degree of disease resistance. And one may sort of argue that they're probably individuals at the end of a, sort of a statistical distribution, and perhaps, they just happen to be old because, they're just at the tail end. But if you sort of look at these individuals, it tend to have certain interesting characteristics. For instance, here is the oldest man that ever lived, doing what he regularly does on a daily basis. And here's actually the oldest individual that has ever lived. And of course, it's very evident what she's up to. But in fact, she's got a stunning resume, and perhaps she sort of gives away her secret when she says she has a kilogram of chocolate every week. But suffice to say, what I'm trying to sort of get at with this particular slide is when we think about therapeutics, perhaps the greatest impact of the CRISPR-Cas systems will be in our, sort of the improved ability to be able to interpret the human genome. And that's perhaps where the greatest contribution of the CRISPR-Cas systems will truly lie. So in that sort of context, I sort of wanted to segue into the second half of the talk, where I wanted to describe some of the ongoing work in our lab in the context of high-throughput biology. And very specifically, how can we sort of start to unravel the human genetic code at scale, which is in a high-throughput fashion. So specifically in this project, what we decided to do was, instead of screening for genes, we wanted to start to screen for genetic interactions. Because it's well known that a lot of systems in biology are often driven by genetic interactions, and often multiple genes are sort of responsible for driving a phenotype of interest. And this project really sort of started off about 2 years ago when I heard a talk by Trey, and it got me very excited, and it sort of got our labs to start to talk to each other. And really, basically, fundamentally, sort of driven by three remarkable individuals, Dongshin, John, and Roman. So focusing on screening for genetic interaction. So there are various paradigms in which we could have done this screening, but the first one that we focused on was in the context of precision oncology. So why genetic interactions important in the context of precision oncology? Well, screening for genetic interactions can really allow us to uncover novel synthetic lethal relationships, which can in turn give rise to new combination therapies. So what do I mean by that? For instance, here's sort of a typical example where suppose you have two arms of a pathway converging to give rise to an essential growth component. And if you disrupt one of the genes, for instance, gene A, then of course your cell will have a reduced fitness effect. But if you also disrupt gene B in concert with gene A, you're not expecting to see too high of a loss of fitness effect as compared to gene A because both of the mutations happen to be on the same arm of the pathway. But on the other hand, if one mutated genes on opposite arms of a pathway that was converging to give rise to an essential growth effect, then one would see this sort of synergy in function between the two mutations and sort of give rise to a significantly greater deleterious effect on the fitness of a cell. And in fact, this has been the basis for a remarkable sort of synthetic lethal therapy, which is now already clinically approved, where individuals with mutations in the BRCA1 gene, which is responsible for double-strand break repair, are unusually sensitive to PARP1 inhibitors, which are responsible for single-strand break repair. So here's sort of an example of where discovering these novel genetic interactions can become unusually powerful. And of course, if one knows the pathway, then it's sort of easy to design it. But often when the pathways are not present, then a sort of a systematic screening approach can actually help us actually infer these underlying mechanisms themselves. So in our approach, essentially what we are really trying to do is basically solve this equation, where we basically want to compute the individual fitness effects of a particular gene, say gene 1 and gene 2, and then also evaluate the fitness effect when you perturb both of these genes simultaneously. And once we have sort of computed these terms, then we can extract this interaction term and then see how significant it is. So to do that, we basically took a very simple-minded approach where we took what I refer to as sort of combinatorial libraries. And so in these combinatorial libraries, basically you have two guide RNAs present on the same vector, but they come in this sort of variety of NC1 plus NC2. What I mean by that is these libraries are themselves sort of subdivided into two parts. You have one where you take a guide RNA and then you pair it with a scramble guide RNA, which sort of gives you the fitness effect of a single gene. But then you also have a library where you have both of them correspond to sort of two different guide RNAs targeting two different genes, and from that you can extract the fitness effect of a double gene perturbation. So one can systematically do this, then one can insert this library into a population of growing cells, and then look at the surviving cells at the end of that experiment and see for enrichment of sort of various library components, and then sort of compute the individual and dual gene fitnesses. So when we sort of started to do this experiment, it's sort of important to realize that from end to end, this cassette is about 1,000 base pairs, which is sort of larger than the synthesis capabilities of most systems. So the way we did it was sort of using the conventional oligonucleotides that can be sort of harvested or annealed or sort of cleaved off from DNA arrays. But the way we went about sort of designing this library was slightly different from existing approaches in that we basically put both the spacers of the guide RNA on the same oligonucleotide. So the pairing of the guide RNAs in this dual guide RNA library was sort of predetermined by an oligo on the chip. And then we took a two-step sort of cloning process by which we sort of assembled everything into a dual guide RNA library. One thing that sort of we very quickly realized when we started this experiment was that these sort of libraries were highly unstable. And this instability really came from the fact that you have a lot of repeat regions on these libraries. For instance, the polymerase 3 promoters are repeated. And similarly, the guide RNA sort of scaffold regions are repeated too. So we spent a lot of time sort of engineering sort of two different versions of the U6 promoter that were sort of non-homologous with each other. And also on the front of the Cas9 guide RNA complex, if you look, the guide RNA is sort of, the scaffold region is conserved, so that is repeated. And so we basically went about creating a library of mutants and evaluating them for their activity. And we're able to find two that showed consistent activity across a range of conditions, which I refer to as version 1 and version 4 scaffolds, which were then used for the screens. So the typical format of the screens was as follows. So we begin by sort of taking a range of cell lines and inserting a CRISPR effector in a safe harbor locus inside the cell. So that sort of gives us a very even keel expression of the CRISPRs. And then we pair it with a combinatorial guide RNA library. And the typical format of results, and just sort of to recap, so we have library elements that give you single gene fitnesses and dual gene fitnesses that can help us sort of solve this equation. So a typical sort of format of results from such a screen is like this. So here was a screen that was done using the CRISPR-I system where you have a crab fused to a dead Cas9. So in the absence of the CRISPR-I, most of the constructs are sort of centered around 0. But in the presence of the crab Cas9, you can see that some of the constructs start to sort of cause a loss of a decrease in the growth effect. And that sort of helps us evaluate the single gene fitnesses. Similarly, one can extract the dual gene fitnesses. And based on these two sort of data sets, one can then extract the interaction term from these sort of screens. But then sort of focusing back on the concept of sort of screening for genetic interactions in cancer cells. So here what we did was we actually used a traditional version of the Cas9, which is good for making double-stranded breaks. And we conducted screens by inserting the Cas9 into the AAV-S1 locus of a range, in a range of cell lines. But I'm just going to focus on our results from 2 different cell lines, just to sort of highlight the concept of precision oncology, like the way we try to simulate it in a laboratory condition. So what we did was we began by taking 73 genes, of which 30 were well-known tumor suppressors, and the remaining were oncogenes. But of the oncogenes, there were a subset of them that were also drug targets. Now that we have a collection of 73 genes, then one can create a single guide RNA library in this dual guide RNA format. And we chose 3 guide RNAs for each gene. So basically, that gives us 9 combinations for every guide RNA pair. And so we have a total of 657 constructs for single gene fitnesses. And similarly, we also did every pairwise combination of targets, and that gives us about 24,000 constructs. So we have a library which is about 24,000 in size. And so using this, we first basically extracted the single gene fitnesses and compared them across 2 different cell lines. So here we compared HeLa versus A549. And already one can see in the single gene fitnesses is that there's definitely some genes that are very cell type specific. And it's sort of interesting to note that P53 has a sort of large gain of fitness effect in A549, but not in HeLa, but that's because HeLas are HPV positive. And sort of it kind of gives us back something that we tend to expect. And similarly, one can also extract interaction scores. And sort of looking at the most significant interactions, here is sort of the network that we extract when we overlap the synthetic lethal interactions in HeLa and A549. And that's basically what is sort of depicted here in this network. And based on this, what we did was we took 10 interactions, 5 of which were predicted to be active in HeLa, 6 of which were predicted to be active in HeLa, and 4 of which were predicted to be active in A549, based on the CRISPR screens. And then we evaluated them in a drug-drug experiment. And basically what we found was that on an average, about 80% of our predictions were pretty good. So we feel very confident about the quality of the data that is coming out from the screens. For instance, here is the example of one such validation that we did, where in the presence of the CHEK1 inhibitor and the ADA inhibitor, you have a significant loss of fitness in the cell types. One can then, of course, start to look at just the interaction networks specific just to HeLa and specific just to A549s. In here, what I have shown in green are the interactions that we experimentally validated, and the interactions in red are the ones that are sort of already found, that were already discovered in literature. So in summary, what you have is a methodology to de novo map genetic interactions and reveal cell type specific shared and private interactions. So in HeLa, we found 52 significant interactions, and in A549, we found 57, of which 12 were shared interactions between both the cell lines. 20 of the 97 interactions that we found to be significant were actually already reported in literature, so that sort of further gives us confidence in the quality of the data that we get from the screens, and of which we evaluated 12 with an experimental success rate of about 80%. So we feel that the precision of our screens is pretty good. And of course, this allows us to now unravel novel combination therapies. But of course, it's sort of important to note here that while we know that the hits that we're getting from the screen are good, there are also a lot of hits that we tend to miss. And I think that's going to be an important aspect that we need to sort of really mine if we have to sort of get the global landscape of genetic interactions, so really sort of map them in an exhaustive fashion. So basically, enhancing screens is a major aspect that we're looking into. And if you have to improve the screens, then we have to start to improve the CRISPR effectors themselves. We have to boost the editing, repression, and activation rates. And similarly, we have to also improve the guide RNAs, not just from the perspective of specificity, but importantly, also pair high specificity with high activity. And what do I mean by that? So this is sort of typical data that comes out of a screen. So as I mentioned, there are sort of 3 guide RNAs that we choose per gene that we wish to target. So in general, you can see that there's typically 1 guide RNA that is very good, shows a strong effect, but there's often 1 guide RNA which is almost dud, which is not functioning at all. And this is kind of important because when we're looking at genetic interaction screens, they sort of scale quadratically. So if you're looking at n genes, the number of interactions are of the order of n squared. And because we're looking at 3 guide RNAs per gene, that gives us 9 combinations. But if we could only figure out 1 guide RNA that was truly highly functional, then you could reduce the library size by an order of magnitude. And that just allows you to sort of screen for a greater number of interactions. So I think this is going to be an important aspect sort of moving forward. And of course, integrating screen data across multiple perturbation paradigms is going to be important, not just CRISPR knockouts, but also CRISPR-I, CRISPR activation, because I think each of these will give us sort of novel insights into the underlying biology. So just before sort of wrapping up, I'm just sort of going to quickly show sort of 2 pieces of work that we've been trying to do in terms of sort of improving the editing efficiency of the CRISPR systems and also sort of improving the efficiency of the guide RNAs. So editing can be modulated by using sort of novel effectors. For instance, here what we did is by sort of using TREX2, which is a known nuclease, you can actually see that across 1,500 sites, it sort of results in a sort of global increase in the targeting rates. And because it's an exonuclease, you can confirm by sort of looking at the signatures of the mutations it creates that it actually does so by suppressing insertions, but by actually boosting deletions, which sort of correlates with the known biochemistry of the TREX2 protein. So one can use novel effectors that can basically boost targeting rates across the spectrum. And similarly, what we've been doing is sort of gathering more and more data based on various guide RNAs and sort of training a support vector machine model that you can use to predict the most active guide RNAs. So for instance, here we took 10 guide RNAs, 5 of which we predicted to be high activity and 5 of which we predicted to be low activity. And in an actual experiment, you can see that the pyogenes, the predictions were sort of well on target. But using another ortholog from Streptococcus thermophilus, you can see that, again, the predictions were fairly on target. And the good thing about the editing experiments was they sort of translate not just in one cell type, but across multiple cell types. So again, sort of our predictive power using the support vector machine model is getting better. So just wanted to conclude by saying that there's a need to strive for efficacious genome engineering if you need to sort of really boost the signal in the screens. So basically have fewer false negatives. And of course, it's important, but it's nevertheless feasible to enhance genome engineering via improved effectors and guide RNA designs. So just wanted to conclude by saying that sort of as we move from reading genomes to interpreting genomes to sort of rationally engineering genomes, I think this is an exciting phase in biology where there's a confluence of rapidly maturing tools. So you have the ability to at least recapitulate human biology with the advent of iPS cells, the ability to put up biology with CRISPR, zinc fingers, and tails, and our ability to assay biology is sort of growing in leaps and bounds with improvements in imaging, sequencing, proteomics, and metabolomics. So with that, I just want to conclude with acknowledgments of my research group. This work was led by Dongxin Zhao in the lab and our really amazing collaborators, my funding sources, and I'll take questions. Thanks.
Great, thanks for that. Any questions? I can start with a question.
Sure.
What is the current limit of how many genes you can look at in combination with your approach and have the assurance that you have full coverage of that set of genes you're looking at?
Oh, how many genes, like in total?
Yeah.
So
what's the limitation of your?
So I think the limitation is more experimental in the sense that as your library size increases, the number of cells you need to interrogate to sort of cover all combinations exhaustively and sort of not lose out things because of, just because of an experimental passaging error or something like that, just sort of really balloons. So what I prefer to do for most of the screening experiments is sort of build small focus libraries.
Look like lists of around 20 genes or something?
Like I would say about 25,000 interactions. Yeah. So maybe, but then the way we create the libraries is you can pair them in a sort of asymmetric fashion. So you don't just have to look at 100 cross 100, but you could look at 10 cross 10,000. Okay. So it's possible to do that. But I think beyond a certain limit, the screens start to get a little unwieldy, and of course your sequencing starts to get even more unwieldy. So focus libraries, of course, it comes with the downside that it's not truly unbiased. So it's guided by some knowledge. Right. So yes, you lose the charm of an unbiased sort of screen, but the interaction screens, unfortunately, because the scale is n squared, have to be sort of pared down to some extent.
One question over here.
Thank you for great talks. I think, have you ever analyzed the actual primary cells or actual normal cells? I think to get a correct and effective cancer target, to avoid the actual interaction of the normal cells, because in case of some small drugs, it affects the actual normal cells. Have you ever tried?
So, I mean, that's a great question. I mean, definitely the need is to move towards more physiologically relevant settings. Definitely like doing these screens in PDX models or something more closer to sort of primary cells is going to be absolutely critical. But it's just from ease of convenience and sort of just from a proof of concept of a new system, we went with sort of regular cancer cell lines. Of course, the dream is to move in that direction, but it also comes with limitations on the number of cells you can culture, how you can culture them, and if there's sort of biases that come from just the fact that you're starting up a primary culture. So, I mean, again, these are, I think, all tractable, but will need some more investment of time.
Actually, thank you. Especially in the hepatocyte, it is essential to avoid actual hepatocyte-specific interaction of a pathway to pathway. Sure. In case of any other cancer, is very effective of this interaction. If such interaction is appeared in the hepatocyte, it's very difficult to obtain the actual target and actual medicine.
No, completely agree. Absolutely. Okay.
Actually, I have another question. Can you think of applying your screening approach to target deconvolution if you have a small molecule, you don't know the specific protein target, or perhaps you know that there's polypharmacology? Have you thought about applying that screening technology to that?
So we haven't explored that, but I mean, definitely, I think I agree. It can be translated in that direction for sure. Yeah, thanks.
Thanks very much.
Yeah.